Hi, our next lab is on photosynthesis. In the photosynthesis lab, we are going to do two experiments. The first one, we will be doing thin layer chromatography on extracts prepared from the chloroplasts of spinach. We'll separate pigments found in the chloroplast of spinach using this technique. And the second part is we will look at photosynthesis in fresh spinach leaves. Let's start by looking at thin layer chromatography. Some of you may have used thin layer chromatography in your other science courses, but as a quick refresher here, chromatography it's a quick method that is used to separate substances in a mixture based on the physical properties of these substances found in the mixture, such as the molecular weight of the substances, their solubility in the solvent, the polarity of the solvent, the polarity of the substances, etc. And in this technique, we use a thin layer chromatography or TLC chamber as depicted here. We also use a medium, a plate, a chromatography plate that is coated with silica gel. And this chromatography plate is prepared by tracing a line on one of the edges of the plate, about 1 centimeter from the edge, a dotted line with a pencil, and this is the origin. This is where we are going to blot the sample from the mixture. And on the other end, that is about 5.5 centimeters from the origin line, we draw another line with a pencil, and we call it the solvent front. And this is the line where the solvent, when the plate is put in the chamber, the solvent will rise all the way to the solvent front. And we use this line as an indication for us when to remove the plate from the chromatography chamber. Now the spinach extract, it's a crude extract from the chloroplast, and it's already prepared for you, and you will use a capillary tube in order to get a sample from this extract. You will dip the capillary tube into the extract, and you will get some amounts at the tip of the capillary tube that you will blot on the origin line on the chromatography plate as indicated here in the picture. And then the plate is put into the chromatography chamber, inclined in this manner, and making sure that the edges of the plate, the sides, do not touch the sides of the chamber, as this would affect the separation of the pigments. The only thing that touches the plate that touches is the bottom of the plate touches the bottom of the chamber, and the top of the plate touches one of the walls of the chamber. But the sides of the plate should not touch the walls of the chamber. And notice also here that the dot where you blotted the sample is higher than the level of the solvent. This is allowed to basically separate for a certain period of time. Usually it takes about maybe 5 to 10 minutes max, and therefore you should always keep an eye on the plate and to observe the solvent as it rises along the plate, and when it reaches the solvent front, the plate should be removed from the chromatography chamber. As the solvent is rising, it's going to separate the different pigments found in spinach based on their physical properties, their molecular weight, their polarity, and etc. And you will get something similar to this, where you can see different bands of different colors that were in the mixture that have separated. Now, how do I identify the different bands, the different pigments? One of the ways is to calculate the retention factor of the different bands that have separated on the silica gel chromatography plate. And the retention factor is actually, it's a factor that is calculated by dividing the distance that the pigment has traveled from the origin, solvent origin, divided by the distance that the solvent has traveled from the solvent origin to the solvent front. So in that case here, the origin of the pigment, basically here, from there to the first pigment is 2 centimeters. So in order to determine the retention factor or the RF factor of the first pigment, you divide 2 by 6 centimeters. And that would give you the retention factor. You can compare this to a reference RF values, whereby the numbers or the RF values were determined using a similar solvent as the one that you will be using in the lab, but it's not always the case. And that should help you with the actual colors of the different bands that you are going to get, will help you to identify the bands or the pigments that have separated on the chromatography plate. In the second experiment, we will look at photosynthesis in spinach leaves, and we will use the same metabolic chamber that we have used in the aerobic respiration experiment. And we will also use the same sensors, a CO2 sensor and an oxygen sensor, and you will put in the metabolic chamber 5 spinach leaves there, and you will monitor over a certain period of time the amount of CO2 that is being consumed or released to the amount of oxygen that is consumed or released using an iPad and SparkView. So that's the first setup. So a setup under light conditions, where you will look at CO2 and oxygen changes over time. You will repeat the same experiment, but this time in the dark, under dark conditions. So the leaves are not undergoing any photosynthesis, and then you will measure any change in CO2 and oxygen level. And you do this in order to get the net amount of CO2 consumed due to photosynthesis. And then the way to calculate this is by subtracting the amount of CO2 consumed under light conditions. You subtract the CO2 produced under dark conditions from the CO2 consumed under light conditions. And here's an example. So the CO2 consumed under light is minus 231 parts per million, minus the amount produced, which is plus 689 parts per million, and that would give you a total of 920 parts per million of CO2, net CO2 consumed due to photosynthesis. The reason why we do this is because while there is photosynthesis happening, aerobic respiration is also happening at the same time, and you want to take that into consideration to get the net amount of CO2 consumed due to photosynthesis. You do the same thing for oxygen produced due to photosynthesis, and you get it by subtracting the oxygen consumed in dark conditions from the oxygen produced in light conditions. And here's an example. Under light conditions, there's oxygen produced the net is 4,587 parts per million. Subtract that from the consumed in the dark, and then you get the total amount or the net amount of oxygen produced due to photosynthesis. Sometimes what you might happen is that the oxygen produced under light conditions is negative, so there's net consumption, basically, and this is due sometimes to the fact that aerobic respiration or the rate of aerobic respiration could be happening at a faster rate than the rate of photosynthesis, and that would result in a net decrease in oxygen. And so in that case here, so the net amount of oxygen produced due to photosynthesis is going to be lower due to this fact, and you calculate it in example 2 shown on screen here.